Let's have a recap. Predict interactions. So we want to predict whether two proteins touch each other. We see that touching proteins, in fact, have different, or the residues of touching proteins have use different biophysics. So in red, you see the interaction between two different proteins here. In blue, you see the interaction between what is yellow here, more over here, between two different parts of the same protein. Green is internal here, and this is sort of expected difference from expected. I showed you that they all differ from expected. I showed you that this is statistically significant. It's not trivial to show that. And I also showed you that, in fact, you can distinguish 6 different types of interfaces, and the difference, again, for all of those is statistically significant. We talked about scientific significance. Think about it. Statistical significance is about quantity. Scientific significance is about quality, qualitative differences. I showed you that in this particular case that also made a difference. Here is an example of what you can do with it. You can predict recall versus precision curve. You can predict from sequence alone whether a particular site is interacting with a different protein. That is the curve here, this light blue. You dial through this curve by simply going from, in this particular case, you have a neural network that has 2 outputs. One is residue is in a protein interface, residue is not in a protein interface. Whatever is the highest value is the one that makes the decision. In this example, in these 2 examples that I show, in both, P is the highest value. Here the highest value is much higher than here. And the way you dial through this curve is by sort of changing this threshold. The more you go in this direction, the more you go to the weaker prediction. The more you go in that direction, the more you have a stronger prediction. Yannick?
Can you go back a little bit and tell us what exactly? So
this curve,
this on the left side with the yellow dots and the one blue dot in the middle.
Oh, and for this particular slide. So this is essentially what I'm showing here is I'm predicting for a set of consecutive amino acids. Shown is a set of 13 in this particular case here. So this cutout here is 20 amino acids. Okay, so essentially you have 1 unit for each one of the 20 different amino acids, and each of those codes 1 position. Okay, so this position center here has 20, or not 20 but 21 because there's a spacer. Let's not get into that now. But so this is 20, 20, 20, 20, 20. So all of them are 20 units. So the entire thing is the number of input residues that you have. In this particular case, 13 times 20 would be the number of input units. Or 30 to 21, okay?
What are the 13 again?
The 13 is so you take a piece of a sequence out, and the sequence reads, in this particular case, DRQGFV. That is the sequence. So this could be, in principle, the first 13 sequences in the protein.
Okay.
But then you slide this window through. So the first window goes for the first 13, the next window goes for residue 2 to 14, residue 3 to 15, and so forth on. You slide by 1, right? And you always ask, what's the state of the central residue? Is it predicted in a protein-protein interface or not? Those are the 2 output units. And you train on a data set. Go through the last slides, through the last talks from last year. So these were what I'm talking about are the talks. I mostly talked about that in December. Essentially, entirely talked about that in December. All the December lectures are sort of bringing this.
What's the algorithm that you designed when you were?
This is in principle. So the part that you're asking really is about a way of coding, and that is not really an algorithm that we did. This is a way of doing it that many people use. I simply use this here to illustrate the idea that you can sort of have, in principle, the neural network creates one answer. One answer means one particular point in the precision versus recall, right? I'm showing you a curve in precision versus recall, which creates an RUC curve or an AUC curve, an AUC value. And you get that by, in fact, looking at different thresholds. At saying, so if you create the higher thresholds, by saying that I only predict P if the strength of this distinction is beyond a certain threshold. Everything else I do not call P. Okay? And this way you get a high precision at a relatively low recall. And then you can have the other extreme. As you see, you cannot go to a very high recall here. In fact, you see that this curve ends at levels that are sort of recall less than 5%. And this is just the problem in this particular case. You cannot do more than that. This is simply where that curve ends. This is the most extreme. So essentially, if you have the lowest possible threshold, you still only get 3% of the true positives, right? This is just the way the method works. So you cannot always create a curve that dials entirely through, right? Is that in principle answering your question? But so for why I show this in the context of predicting protein interaction site, go to the slides or to the talks from December, essentially. And it's important because this is a completely different topic than the one that I had before. So with subcellular localization, you always use an entire protein sequence. Now you sort of slide a protein sequence through. So many people in the room here had the lectures on structure, and we used mostly this kind of philosophy. But in the course of this lecture, we have actually mostly used something else. So in this course of this lecture, this is the first time that I'm sort of going through. I will bring it back again next Tuesday. For what I continue to do today, I will also go back to entire protein. But next week I will get back into this because we want to predict effects of variance. And effects of variance are really, again, we sort of look at the effect of a variant in context of what is next to it in sequence. And principally you would want to do what is next to it in space. Because what happens to a protein is always relevant, or is always depending on its contacts. But you don't have that in all cases. Always have sequence. At least that's the assumption of these methods. Can I keep it there? Okay. So this way you dive through that curve. You always have to compare any method that you have to random. There are different ways of creating random. And it's always a challenge if you develop a method to come up with something that is not quite random, but it's something that you say is a dumb model. So if you want to predict protein interaction site, what could be a dumb model? It's not random, something possibly better than random, but it's something still dumb in the sense, so my definition of dumb is you can think about it, you can code it in less than half an hour. So creating this neural network, collecting the data, creating the network, that takes a long time. Long time on the ballpark of master thesis. Okay, this is not, in this particular case, this was much more than a master thesis. It took over a year. This was on a ballpark, but there was a lot of things that had to be done before. And like very often, we did not expect that it would take that long. But it was also something that nobody ever had done before. But then on that scale, so you spend a few months on your method, and you want to sort of compare it to something, there's nothing else out there, and you want to compare it to something minimally or really bad enough. At least you have to be better than random. Flipping a coin, okay? Again, doing random you can do in different ways. But the second thing is, what could you do in order to sort of do some dumb thing that is possibly better than random? Can you sort of come up with a simple thing that you could immediately code? If I gave you the data, at this point you have the data. Yeah?
My question is, what is random for you? Do you mean like already considering data imbalance or such?
Yes, so again, random you can do in different ways, right? Yeah. So I'm looking at the positives. So most residues are not binding. Say 10%, I'm not sure what the number here was. But say 10% of the residues bind, okay? So if you always predict every residue not to bind, and if you measured precision in terms of Q2, so overall number of correctly predicted residues, you would have 90%, right? It's something, a totally stupid prediction, always says not binding, getting in Q2 and 90%. And precision it would not get 90%. So it depends a little bit what you plot here, what is relevant. So here random was reshuffling. Simply reshuffling the labels. Keeping the distribution, so keeping the imbalance. Again, there are different ways in which you can swapping the labels. There are different ways in which you can do that. But my question is, what could you come up with that you at least would believe is better than random, and you would believe is something that is simple enough to program in a very short period of time? Yeah? Oh, no, it's just, yeah?
What we always do amino acid compositions?
Yes, so that is another way of doing it. So in this particular case, again, you're threading a window through, so amino acid composition. So one way of looking at that in terms of deep learning is one-hot encoding could be sort of a background model. Because you have the data here, and you sort of have a clever way of using the input. One-hot encoding is always the simplest, most stupid thing you can do. That is, but in this particular case, so something simple. Amino acid composition only. Well, again, so you would predict for an entire protein where you look. But other than that, if you
say leucine has a 20% chance.
So here we are. So you look at some biophysical features, right? You say binding residues most likely are somehow residues that are somehow hydrophobic. And I want them to be exposed. At this point, maybe you have a prediction for exposed, or if not, you can do a simple one by some average hydrophobicity index. And then it has to be exposed residues, maybe exposed residues that are somehow a little bit hydrophobic or something. And then I call them glue. And you can see how well you do, right? And this is something that everybody in my group has to do just before they publish. And in some of those cases, we actually discovered that a very dumb method, so this is sort of, in some sense, is the worst case and best scenario. So you have worked on a method for 6 months, now you come up with a dumb method and the method beats you. The dumb method beats you, right? So this is bad because it's really complicated to publish. On the other hand, at least you can publish. Listen, everybody has always done that. I can beat everybody, but I'm actually not better than dumb. This is sort of, we just have written up a paper like that. It's not a paper, it's a manuscript, and we hope it becomes a paper. It's not that easy to get those through the reviewers. On the other hand, it's frank, and you should try to do something like that. I showed you that the big improvement over what I showed you so far is by including evolutionary information. In evolutionary information, again, I have my protein sequence, I find sequences that are related to it. In this particular case, I have 4 related sequences. I align them, and rather than putting the one-hot encoding into each one of these 20 things here, so here, each one of these bubbles here is 20 symbols that are essentially a binary vector, because it's 1 if the amino acid there is that particular amino acid, and it's 0 for all the other 19 amino acids, right? In that sense, it's a so-called one-hot encoding, a simple binary vector, a simple vector that has 1 for 20 different amino acids for each of these bubbles. 13 times 20 or 21. And now you replace that by something that is no longer binary, that's a real value. And the real value could, for instance, be the number of times you see the D in that family. So in this particular case here, again, it would be back to the binary coding, because the D would get the entire signal, and all the others would not. In this particular case of a D here, you would see that the D gets only 20%, while the N gets 80%, and the Q gets also 20%. So essentially you would have an average over the number, the residues you see at that particular position. That's all you change. Other than that, you keep the same device. And by doing that, this curve is not quite only that, it's a little bit more. You also use secondary structure, solving stability, and some other features. But you see that now you get a curve that you can actually dive through a variety of levels of recall. And at any level, it is clearly higher than the one from sequence only, so you gain a lot. I also showed you that we can look at the strongest predictions here, and they are related to hotspots, and we can use that to predict hotspots. Now, one problem that we have with protein-protein interactions is we get these hairballs. You may have seen this on this level, where you just have a bunch of interactions. You cannot interpret what that means. If you are lucky and you can really put that into pathways, you see that protein A activates B and activates C, and you build the whole pathway. Here's a pathway. There's a temporal resolution. There's an action resolution. We are sort of trying to get at yes?
Was that made by the output of the graph?
No. So this is, in fact, the way these, ultimately, it is getting more difficult to make these. So historically, the way these are done, and I'm showing here things from the KEGG pathway. KEGG pathway essentially was based, so that began, wait a minute, I'm sorry. That began almost 30 years ago now, 92, he began to collect. And the way it was simply done is by careful one-by-one experiments of testing whether A binds B. Many experiments had been done to test whether A binds B, has been put into the literature, and a group of people simply took this literature and annotated these pathways. That's KEGG. It's 30 years of work, hundreds of people that have worked over 30 years, not integrated, but every year hundreds of people work on building this KEGG pathway. Essentially they take the entire literature, right? That's the historical way of doing it. The new way of doing it is you have large-scale experiments, and each one of these experiments is not giving you a clear answer that A binds B, but it's a probabilistic answer that A binds B, binds C, binds C. So it's a probability for binding a bunch of proteins. Typically tens or hundreds or even 1,000. You get a probability that A binds to 1 of the 1,000, something like that. But that means you get these big groups, or these big hairballs, or these big connections, and to take them apart and get into that is highly non-trivial. But that's what people do manually, okay? Again, one resolution is time, so you measure that the interaction happens at different times. One resolution really is where, localization, and then also to essentially build up these things. But in order to do all of that, we would need networks of interactions. And how can we get networks of interactions? So one idea here, historically, the first way people did that is through microexpression correlation. So the idea really is, if two proteins are correlated or expressed in the same way, then maybe they interact. Because they are co-expressed because they have to interact, right? This is sort of the understanding. Is it true or not? Well, when we did this work, and so that was done by Tarzan. So when Tarzan did that, there was a lot of literature that said we can use co-expression to predict protein interactions. Somehow we simply tried to sort of repeat that. The largest ways to aggregate data about interaction were then still are used to hybrid and affinity purification. Largely those are still the largest things, except for this is now much faster through high throughput sequencing. So this is essentially to see what glues together, and here you sort of test systems to really see individual pairs of proteins. We see some correlations between microarrays, but what we actually see is that microarrays reveal functional associations. They don't show interactions, so they do not really show A interacts with B, B interacts with C. They just give the whole thing here. While we really are talking about these pairwise interactions. And that is not what microarrays give. And the question then became, can we actually predict physical interactions from the microarray data? So using it, just take a machine learning device and go ahead, didn't work, or just use it like that. So what we did is we combined a lot of data sources, we put the microarrays in, and things got worse. We got the microarray data out, our prediction was better. And that is the first way we realized that microarrays are sort of a little bit tricky. But now the question is, can we sort of learn to use the microarray? And there are essentially two ways to do it. First, you have to sort of group the microarray data, and then you have to put some machine learning device on it. Grouping is noise reduction. I'm going to skip through this very quickly, because ultimately the message is you can use microarray data. Just to make this point again, I want to go through too many slides today, and there is something else that I find is more important. So that's why I'm zipping through, and I see that time is somehow already advanced. And I cannot put it into the Tuesday lecture, because on Tuesday I'm going to be busy too. So I believe I'm realizing that the semester is going to be over soon, so I'm speeding up a little bit. And again, it is my mistake that I didn't do this earlier. It's not your mistake, and you're not going to pay for it. So this is not things I skip over like this will not be part of the exam, right? Just to make that clear. You can train on it. By itself you cannot use it, but you can train devices that essentially use microarray data and also in fact mass spec data to really predict pairwise protein interaction. Yes, Paul?
I think that, so that what you're getting at the top, that doesn't this tells you just maybe like chains of interactions and not individual interactions?
Yes. Which is essentially what I call associations, right? And is, again, so most of the literature uses associations because for functional associations or for functional studies, associations have a lot of importance. So, and a lot of these hairballs are created by associations, not really by people predicting again, if I had all the data of all protein interactions, the hairballs of real pairs would still be two hairballs. But what we have today mostly is because we are essentially looking at associations. Again, we are looking at anything that is somehow related as interacting, and in some sense it is, right? It is associated, but it's not interacting in a physical way. And physical way is relevant again in the context of drug design. So one protein, so I have a variant of a protein, and that in some people causes a problem or not. That has to do with what it interacts with, and whether that one it interacts with is sort of having a variation. That is ultimately the cancer study. What we realized using these associations alone did not really get us to understanding cancer, or gaining network biology to doing better on understanding cancer or curing cancer or helping patients. So part of that may be that we are missing the component of physical interactions. And part of that clearly is that we still don't understand how to best correlate the data, and we don't have all the data. So there's a bunch of challenges, but that is one. Okay? We can in principle do that, but we have to sort of train a machine learning device on that task. Now let's get into a few issues about learning, machine learning, artificial intelligence. Over the course of my lecture, I'm sort of sitting, unfortunately, still in history. I keep talking about machine learning. Today we call that artificial intelligence. It's the same thing, it's just that the language has changed over the last 2 years. And I've not been fast enough to change all my slides, or my head. The point that I want to make here is prediction clearly is the acid test of understanding. So if I believe that I understand the deep down of scientific problem, I should be able to predict. Right? If I'm not able to predict, I don't understand. That much is clear. What about machine learning? Does that extract understanding? I can predict, but do I get understanding? So the first step that I want to make here is the world has changed from 1997, at which for the first time a computer beat a chess champion, Garry Kasparov, to the situation in 2016 where a computer beat a Go champion. The reason why this is a different game really is because this is way more complex. Why I say that the world has changed is essentially this here. What is the difference between these two?
I would say brute force versus intelligence.
And that gets back to, this is a solution, brute force. You simply look at all possible alternative ways to play the next game, and the bigger the computer, the more steps ahead in time you can do. That's why Deep Blue, at that very point in time, not in 96, but in 97 could win. Because in 96 it was still too small. It could not go ahead enough. It was not a mistake in sort of assessing what is a good move, what is a bad move. That was not the point. It's just the number of steps it could do. That was the big gain, and that sort of made this win. Now, this is historically, if you think about it, always the way we have been thinking about computers. They are these big muscle machines that can do simple things very quickly. The same thing again and again and again. The world has changed. So this is done by artificial intelligence. But the important point is not only that this is done by artificial intelligence. The point is that today, on your machine, you will not use this idea. Your laptop that you have in front of you is bigger or faster than Deep Blue. But if you have a chess game on there, it will not do that. It will have a program. There are these simple programs that you can get. They use artificial intelligence. Why? Simply because it's faster. It takes much less time on your computer. You get the same result for much less. And that is the shift here. The shift is that today, now, over the last year or two, people now begin to, in fact, reprogram solutions they have. They They are completely accurate solutions. For instance, one above here, they do autonomous flight. So there are certain mappings. You have the exact solution, but you're not using it because this little quadcopter is so small that you need less to put the CPU. You have less CPU or GPU, whatever you need, and you can do that with artificial intelligence. So you reprogram all the right solutions by approximation simply because they're way faster. So the way we think about computers is changing. That is the big shift. It is still also true that the types of problems you can do with machine learning are different ones. Now, historically, people have sort of argued machine learning, artificial intelligence is sort of these black box machines. And this is absolute utter nonsense. This is something where people completely misunderstand. And that comes ultimately from people trying to extract the rules from the black box that the black box learned. And then they got to rules, such as for our device here that predicts whether particular residues in the protein interactions or not. It says that if it is 1% or 5% conserved, if it is an alanine or a C that is 3% conserved, or a G that is 5%, and if both are 2% in a second, and it continues going. And you're already beginning to lose me. But the kind of pattern that you get from 13 adjacent amino acids, for which you then also have conservation patterns, secondary structure prediction, and, this is way more complex than that. So you write out the rule, an expert looks at it, I don't get it, right? So this is not a reasonable rule, and that is the mistake. The problem is, the problem we are applying here is so complex that we cannot understand it. So many of again I said there's a philosophy change or a perspective change, so we're now applying machine learning to things that we can solve, but we are still applying it to problems typically that we cannot solve with a simple equation. And when we cannot solve it with a simple equation, the machine learning is going to match the complexity of the problem. There are no simple rules. When we look at the machine, what the machine learning has learned, it's complex. Because the problem is so complex that we don't have simple rules, all right? And that any of the simple rules, they don't get the same performance. That's the sort of simple method that I said you should invent, your dummy method, that typically is much worse if you do the machine learning right. So it is not a black box. It really extracts what I would argue is understanding. And in some sense, in life sciences, is the modern understanding, yes?
I mean, also, even if you want to pull out more human understandable hypotheses, you can just do that by testing.
This is in fact...
It's a different type of way of testing. You just change your input and see how the machine learning reacts and pull... True, Paul. Out of that. True, Paul.
And that, in fact, is what today people refer to as understandable AI. And it's something that Google or all the companies are highly involved in, and we all will sort of benefit from that, or will spend some time on that. Anybody who applies AI over the next years will come in contact with that. We have to do that, and part of that comes from sort of processing the data in different ways, so looking at the data in different ways. I'm going to show you some examples. Underlying it are 2 points. It can extract understanding if and only if you do the cross-validation or whatever you call that process right, and if the data set that you have is representative. So representative means simply you want to predict the future, and if you have a slice in the data today that has nothing to do with the future, you cannot do it. You cannot. But as long as the data today is representative, and as long as you do that right, I believe you extract it. Understanding. So the first thing about cross-validation, you have a data set, you split it into 2, one you hide under the table, and you see how well you do. This split cannot be done at random. You have to sort of understand again the correlations in your data in order to say how you split the data. For us, for instance, you cannot have protein from the same family for training and testing. So in the field of proteins, this is, in some cases, relatively easy. This is not enough. I told you we need 3 data sets, but before, in fact, we need to have every single protein taken once for testing. So we have to somehow find a way to rotate through different model extractions. And then we need 3 splits. So training, cross-training, and testing, or whatever you call it, that is such that anything that is sort of model building is here in training, and anything that has to do with hyperparameter optimization, model selection as they typically call it today in a more generalized way, is what I call as cross-training here. And then there's testing. And testing happens if and only if the whole thing is done before. So you have only one model that you test. Only one final thing that you believe is the right solution, and that you test in comparison to other methods available, but not to other options of handling it, of other models that you created. This is sort of the difference here. And if that is done, then it's still not enough. You try to get a pre-release data set, so a data set of proteins that have come into databases while you were doing all this development. And if you do that all right, then you get a paper that's sort of cited very often, but you get a paper that in fact states a number that stays there for 30 years and is right, okay? If you do that right. Now if you do the same, so I was involved in the team sort of who got it all right here, and I was also involved in the team who got this one here. And we did exactly the same thing. Except for, there are many co-authors, I just don't list them because of this slide here, except for this was totally wrong. And the reason why it was wrong ultimately was because the data that we had available, the experimental data, was not as good as we thought. The experimental data was crappy. But again, this is our mistake, and this is why that thing here, in fact we published this later, saying this was crap, and this was very, very complicated to get through into the literature. So this issue here, so it is not always enough is the conclusion. But one thing, if done right, this cross-validation and data preparation, then we can sort of show that we have understanding. And that is this story, Janai Ofran is the point here, is the person behind, because it's his project, where simply the stronger one here tells me about hotspots. I simply use the strength and the prediction to define hotspots. I never use in my training, in my testing, anywhere the idea of hotspots, yet we can predict hotspots. So the system from the data extracts some data, some reality, and that is some understanding. I can predict hotspots. Okay, it's one understanding. I understand how hotspots look. Well, the machine understands it. We are back to the rule story, but the machine acquired some understanding that I did not put in, because I actually didn't even know it. Now, we can clearly gather understanding, and we can also completely do things wrong. Here's an example where we simply want to predict, and that's the story of today again, so let me get back to that. Now I want to predict that this protein binds to that protein and not to some other protein. So I have two proteins A and B. I want to predict that they bind, not where they bind, that they bind, okay? And we sort of went through an iteration of, yes?
That does include the one task, the other one?
That's what we thought. So we first had a method where we predicted where it binds, and then we simply decided on that it binds by, if I have predicted enough binding sites, then I call it where it binds. And then we created another method that just did the binary thing. And for the binary task, the other method was way better. So then we decided that we need two different methods, and we do it one after the other, and that order of events is right. That is all I can tell you. So there is no, if you think about it, you would ask your question, and and I cannot tell you why. Ultimately, the reason, no, I cannot tell you why the answer in practice is no. My feeling is the answer is no because we don't have enough data. If we had enough data, most likely we could just have the same solution in one go, in one deep learning device. But currently we still don't manage, is ultimately the question, the answer. Is that enough for you?
Yeah.
Okay, so the but here was twofold. We can do it, but first part of but that is a problem, method 41. So it took a long time to get to a method that worked. But then, you know, the great thing is that method was extremely good. Very, 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 very good. It had a sort of tiny bit of a problem. Let me illustrate the problem. So what we did here is, as positives, we used PDB. So proteins for which we know in 3D structure that they interact. We know where they interact. This is not what we use here. All we use here is they interact. If I have a crystal structure of a pair of proteins, then I know they interact. Okay? Very clear. It's a very good very good proof that they really interact. Then I need negatives, so proteins that don't interact. Let me go one step back. I have to do this for one particular organism. So I'm going to do that for human here, right? Now, PDB is the highest resolution data set for positives that I can get, so I take that for the positives. For the negatives, what do I take? Well, people don't measure that two proteins don't interact, or they do measure that, but they don't publish it. Or very few publications say, I have measured this, and this is again obvious because people don't dare to claim that they don't interact, because maybe there are some conditions they didn't try. Maybe A and B interact, or A and C interact, but they just didn't try those conditions, so they're not going to say they don't interact. They can only say, under the conditions we tried, they don't interact. But this is not a message that people want to read. This is why experimentally we find little. The way now we did it, we argued, if nobody, by whatever resolution experiment, yeast-2 hybrid, mass spec, whatever they have, they say that A and B interact, we throw that out. Positives are only the ones in PDB. Negatives are everything that nobody in any resolution has ever seen. Okay? Is the idea clear? So what's the problem? So there's a small problem and a big problem. The big problem we didn't foresee. What's the small problem? Yeah? Or whichever.
Well, I mean, people don't test every single combination.
Yes, that's this. Well, let's get a step further. That's this. What I would call the small problem. There are many mistakes in here, but it's even sort of more relevant here because if you think about it, I want to build a method that predicts interactions for proteins that I don't know. So every case of a protein pair that I want to predict actually sits in that data set. So that makes this small problem a little bit bigger, but it's still a small problem. The real big problem is a different one. But we have to understand that. So we are trying to use a negative data set that is exactly the opposite of what we're trying to have a method that learns to not always say no to the no's. Okay? This is sort of a built-in frustration if you want.
You have interaction proteins in your data set that were just never tested.
Yeah, that's what I'm saying.
Yeah.
Well, actually, this is a little bit more complicated. So we cannot put proteins in here that have never been tested in any way. So those we skip. We have to have proteins for which somebody at least said it binds to some other human protein.
Right. Combinatorically, you're not doing it.
I'm not doing the 20 by 20. Exactly because of the reason. So things that nobody has ever touched, I cannot make a statement about, right? Or let's put it like that, that would be sort of very daring. In fact, it's getting slightly more complicated, and this is sort of the first issue that we had to address. We can have the negatives only as a subset of the things for which we also have positives. So in principle, these could end up being completely different proteins, the negatives and the positives. Because some people have done a small-scale study on something for which we never saw in the PDB. And that actually is also some issue, but let's not get there. The big issue is a different one. Can you sort of... We could not think about that. We could not predict that. So I want to hear from you something that I never expected after 20-something years in machine learning or AI. So I expect a lot from you. What's wrong? So we actually created a method that predicts whether a protein is in the PDB or Swissprot. So essentially, this method learned. I did not know that. We know that proteins in the PDB are shorter than proteins here. Yes, but that you can really look at a protein and predict that it's PDB or that it's Swissprot, I did not know that you can do that. But this method here clearly learned that. So method 41 was a fantastic way to predict whether you're from PDB or Swissprot. It has some sort of limited relevance, right? You could have just looked up the database record. That would have been a little bit easier. So this is an example where the dumb method is way better than yours. But, you know, the real problem you immediately see is something else. The real problem is that we created a solution that zoomed into something that we absolutely did not want it to zoom in. It never learned anything about interactions. Yes?
Did you figure out what the difference between the PDB?
What would you do? How would you solve it? How would you solve it?
You could look at some properties.
Yes, that is not easy. So length is the immediate one, right? And the moment we look beyond length, so these are shorter than these, but not all of them. And the ones that are not shorter, so it was not that easy to go much further, and we did not want a method that now explains why we can predict PDB, because you're sort of getting, you hit your face against the wall, and so now I jump against it and I make a story out of it. It's not so much fun. But ultimately, we have to find a way around it. So what's the way around it? You had another question, or?
I wanted to ask why they're longer in the system.
That is sort of getting to your question too. So what happens is that you say you have a protein that sort of has this shape where here's a sort of floppy. Okay, for floppy I would have to. I don't have anything floppy now. So you imagine this to be a cable, and this is floppy. And I cut this away before I crystallize it, simply because the crystal is much easier to build on something that is more compact. Okay, so proteins, anything that sort of sticks out is cut out in order to get a PDB structure. So by that process already, on average, they are shorter. The next thing is, in some cases you have proteins that form two domains, where this domain is easy to do a structure for, this is not, so you cut that one out. So it's a selection of the subsets that are easier to form crystals. That is a selection of shorter ones. The shorter, the easier. Typically, there are exceptions to that. Put it the other way around. So every protein of 1,000 residues is more difficult than any protein of 200, or something like that, residues. So on the extreme, you can really see that this completely holds. And that is partially, sort of, in some sense, the understanding of the difference. But there's a little bit more to it. The hack to avoid that is now, instead of all you take the PDB for is the lookup for A binds B, okay? So you simply base that now on the sequences from SwitchProt. You're not actually using the PDB sequences, but you're using for the positives and the negatives the sequences from the same database. And just take the label from the PDB. Which, by the way, is another reason why we have to split these two methods, because the moment we predict where it binds, we need to go back into the PDB. We cannot do it the other way around. We cannot do whatever I don't see in PDB is not binding, because PDB is just a small subset of all possible binding interactions. Anyway, so the second major step here is the preparation of the data set, and this is going to be a very, very fast through. So the one issue here is about grouping. The way we cluster into families for protein interactions gets more complicated. The first story of complication was published first by Yongki Park and Edward Marcotte. They distinguish three cases. Case 1, case 2, case 3. These cases are depending on the overlap between training and testing sets. Say, you want to test on whether A and B bind. And if you used in your test set A binds C and B binds D, where C and D are different from B, okay? So essentially we are in the training set, we have an interaction between A and something else than B, right? That is class 1. In class 2, one of these two you have and one of them you don't. In class 3, you do not have any of those. So if you want to test that A binds B, you will never use A for training and never B for training. Mind you, what we're actually training on is not A or B, we are training on the interaction. So it's not evident that this would make a difference. In fact, before this publication here, in 2012, nobody had ever distinguished that in any context. And they find that the performance decreases. So this case where we completely discover new interactions is the most difficult one. Okay? So we do really have to distinguish this, and this sort of shows that in more details. The next issue is, do we have to do the grouping on the positives and negatives? And the answer here is on both. So this is the first time that we learned that the redundancy reduction has to also be done on the negatives, although the negatives somehow are simulated. I still say everything that has not been observed, which is not really observed, right? And on those data set, I still have to do a redundancy reduction. And that gets us into sort of doing redundancy reduction in networks, and that is really, really not easy. I mean, it's relatively straightforward, but it's just tricky, complex. The next question is, do we have enough data? How do you typically, is there a way in which you can answer the question whether the data that you have is enough, are enough? Yeah?
If you remove some, you lose performance?
Yes, exactly. So you have the data set, you see that the performance is much worse, you see that you extract that most likely more data will help. You have the data set, you get the same performance. Well, you know that doubling the data will most likely not help. That could be either because your model is not good. It could be because your data is not good. But at least you have some saturation that you see already. So clearly, this is not immediately crying out for more data. So if you see half the data gives the same performance, the first thing you do is reduce the parameters and see whether a smaller model will still be good. And if you still see that, then it indicates that there's something wrong with the model or something wrong with the data, or you, in fact, certainly the issue clearly is not that you need more data. But one question that we had using all the data was, say, I have again protein. Again, I will neither. If I test A and B, neither of those will be in the training. But still, the question: if I have in the training a protein interaction between A and B and C, and B and C are very different, should I use both, or should I use only one? Now, in our particular problem, we knew we have an issue with data set size. We don't have enough data. So we thought that as long as both of these interactions, as long as B and C are different, this is a different interaction. Going back to the physical model, it could even be that A interacts with B at a different site, on a different side really. So the interaction between A and B, so this is A, this is B, could be, and this is C. It could be like that. So I would not have anything to learn from knowing AB about A and C, right? Still, the result was I should use every single protein only once, because alternatively what the system immediately zoomed into is saying that there are sort of multi-interactors, and I just give a higher probability to predicting them, no matter what. So the network immediately zoomed into predicting multi-interactors the moment we put this in. Yes, thank you. I can also do it here. It's a little bit further removed from you guys. Thank you.
If anyone is cold, just no, it's really hot. And I was also
I'm sorry.
thinking about
So ultimately the point here is, and I apologize, I'm really fast. So the problem really is more complicated here. But now we get into the issue, how do we do it best? This somehow is an animation that gets back to Yannick's question, and given time I'm not even going through this, so I'm not testing, I'm not testing all of these. I put it in a grinder and I simply say a binary decision. And what would you do as a binary decision method? What method would you use in order to make the decision A binds B, just overall, an overall signal? What kind of method would you use for that? What would you use as input? Yes, I'm so fast that I completely lost you at the get-go, yes?
You mean the method, like neural network, or
what type of input signal, or what kind of thing? Yes, something like that.
So
is there anything in this lecture that you have encountered that may sort of be interesting for this task? I want to predict that A binds B. Yeah?
The amount of amino acids.
The amount of amino acids? What do you mean by that?
The ratios.
So essentially amino acid composition. So we did that amino acid composition. We threw in some other information such as solvent accessibility, predicted signal structure, some biophysical features. We threw in the fraction of amino acids predicted to bind to other proteins. A bunch of these features. We created something that had ballpark 100 input units, and we could do the job somehow. But then we improved much over it. How? By something that we have already encountered in the course of this lecture. Any idea?
You define the data set or increase it?
Yeah, no. Yeah. I mean, as a method that we use for predicting something else over the course of the last few months. Yes.
Isn't SVM from TrueLog?
Yes, exactly. The binary decision is done by a proper kernel SVM, and the proper kernel SVM essentially, again, to go through this method, essentially what it does, it finds conserved k-mers. So k-mers that are conserved within a family. And then it compares sort of matching k-mers. So I may have told you that for log3, we used this concept, predict subcellular localization, and the subcellular localization sort of used about 1,000 k-mers, or 1,000 plus, but ballpark 1,000 k-mers, right? Now, since we have a sort of square event, we have 100,000. So this is a huge input space for just a few thousand samples that we have as positives. And by the way, there is also an issue about the negatives, so the ratio between positives and negatives. It is completely imbalanced. We sampled it at an imbalance of 1 in 50. So for every positive, we have 50 negatives. And we do not know whether 1 in 50 is the right solution, or 1 in 100, or 1 in 10. Experimentally, it is unclear whether it's really between 10 and 100. So we just picked a number somewhere in between. And this is ultimately what this one says, and I'm just going through it.
How did you do it then? Did you cut proteins? Or how did you get a ratio?
The k-mer?
Yeah. How do you get a ratio of 1 to 50?
Well, no. For the ratio of 1 to 50, what I mean simply is the mix of positives versus negatives.
Yeah, but
so
if a typical protein has 300 positions and only...
Not positions, we have only A and B.
Oh.
We're not looking at positions.
Well, why did you do half-half?
Because in nature we know it's not 1/2, and it's not half-half. So then the method would completely overlearn interaction, would completely overpredict interactions. Most proteins, so ultimately the idea is that we then applied the method to 20,000 proteins, right? So now once have gone through all of that, I'm going to predict for every single human protein what its binding partners are. If I had sampled at 1:1, most likely for every above some threshold, I would have 10,000 likely above a threshold. While sampling at 50, I have sort of 200 above the threshold, or 400. I'm slow. Or something like that, right? Which still most likely is a high number, but 10,000 is a higher number than 400. Anyway, so one part that I want to still point out, C1 here is the example where training and tested, the test protein has been used for training, but not in the interaction. And what you see is the blue is higher than the light blue. So blue is our method using some data from expression, light blue is from expression arrays. And expression arrays in both cases do not really help. So light blue is not much higher than dark blue. To put it mildly, you may also say it's lower. But at least here for the C1 class, there's a little bit of a region where the light blue is above. So expression array somehow helps. This is expression array just as a lookup, not expression array as I showed you before that you train a machine learning on it. And I believe this is where I leave it for today. This simply says that we can also predict where it binds, but the method is so unclear that we still have not published that. Where things